So I'm going to start off with a bit of an introduction to the digestive system. Now, before you watch this PowerPoint, you might want to watch the video link, BBC Bite Size. It's just got a clip about the digestive system, which is quite good. Okay, so looking at the structures of the digestive system. So the salivary gland would be found at number 2. And then number 1 is just a mouth. You've got 3, which is the esophagus. Number 4, more part of the breathing system, but number 4 is the diaphragm. Number 5 is the stomach. Number 6 is the sphincter, which will periodically release some of the chyme, some of the mushy liquid out of the stomach into the duodenum. Number 7 is the liver. Number 8 is the gallbladder. Number 9 is the bile duct. Number 10 is the pancreas. Number 11 is the duodenum. Number 12 is the small intestine. Number 13 is the large intestine, the ascending limb, because it's going up. Number 14 is the appendix. 15 is the large intestine, but the descending limb. It's going down. 16 is the rectum. And 17 is the anus. So looking in a bit more detail about what each part does. So first of all, in your mouth, obviously mechanical digestion with the teeth to mush up that large chunks of food into smaller pieces. Number 2, the salivary gland will release amylase, which will start to break down any starch in the food into glucose. Okay, then the food will be pushed down the esophagus by peristalsis, where the smooth muscle will squeeze the food down into the stomach. In the stomach, there is hydrochloric acid, which is acidic, and also pepsin, a type of protease which will break down proteins into amino acid. So hydrochloric acid is actually the optimum pH for pepsin to work. The stomach is also surrounded by a mucus lining to stop the hydrochloric acid from, digesting its own wall. As we said before, the sphincter will let out some of the chyme, some of the fluid, some of the churned up food from the stomach into the duodenum periodically. Number 7 is the liver. So here is where the bile is produced. Number 8, the gallbladder, is where the bile is stored. Number 9, the bile duct, is where the bile will be released into the duodenum. Number 10, the pancreas is where amylases, proteases and lipases are produced, and they'll be released to act in the small intestine. Just a note about bile. Bile neutralises stomach acids, and it also emulsifies lipids, which means it breaks large fat globules into smaller fat globules, which then provides a larger surface area for the lipase enzyme to act on. The duodenum is just the part of the small intestine between the stomach and the main bit of the small intestine. Number 12 is the small intestine. So in the small intestine, the final bits of digestion take place. There are membrane-bound enzymes such as amylases, lipases, proteases, which will do the final stage of digestion. They'll convert proteins into amino acids, lipids into fatty acids and glycerol, and starch into glucose. And those small glucose, amino acids and fatty acids and glycerol will now be small enough to fit through the microscopic holes in the villi, the small intestine wall, so that they can be absorbed into the small intestine. We'll go into a bit more detail about how they're absorbed later on. 13, the large intestine. So the large intestine is where the water is reabsorbed from the food to leave behind solid feces. So any water is reabsorbed back into the body so that you end up with solid feces, which is then stored in the rectum until it's ready to be egested. Egested is the opposite to ingest. So when it's ready to be egested, it leaves the body through the anus, which is number 17. So just looking at the role of enzymes in digestion briefly, and I'll go into more detail about this later. So there's two stages. The first stage is physical breakdown, which is your teeth breaking down large bits of food into smaller bits. And then you've got your chemical digestion, which involves enzymes. And looking back at GCSE, the three that you need to know about is lipids are broken down into fatty acids and glycerol by the enzyme lipase. Proteins are broken down into amino acids by the enzyme protease. Starch is broken down into glucose by the enzyme amylase. So physical digestion, the large food pieces are broken down by the teeth. This provides a larger surface area for the chemical digestion for the enzymes to start working on. Also, there's a bit of a physical breakdown in the stomach where the muscle wall will contract and relax to perform a churning action. Then you've got your chemical digestion. So the definition of digestion really is to break down large insoluble molecules into smaller soluble ones so that they can be absorbed into the small through the small intestine into the blood, and then they can be assimilated by the body to use elsewhere. So assimilated means making bigger molecules in the body to make useful things. So the function of digestive enzymes is hydrolysis. If you split that word up, hydro means water, lysis means splitting. So hydrolysis means the splitting of a molecule using water. So you break down a molecule using water. So hydrolysis is the splitting of a molecule by adding water to the bonds that hold them together. So sucrose, if you add water to sucrose, it splits it up into its two monomers, glucose and fructose. Just to note that enzymes are very specific, so one type of enzyme will be needed to break down larger molecules, and another enzyme will split the large molecule into sections, and then the sections will be hydrolyzed by a different enzyme. So the three types of digestive enzymes are carbohydrases, which break down carbohydrates into monosaccharides, lipases, which breaks down lipids into fatty acids and glycerol, and proteases, which break down protein into amino acids. So some examples of where these specific enzymes work. So amylase is secreted in the salivary gland, and it's a type of carbohydrate which breaks down starch into maltose. Now maltose is two glucose molecules together. Pepsin is a type of protease which is produced in the stomach, and it breaks down proteins into peptides. In the pancreas you get quite a lot of enzymes. You've got amylase, which breaks down starch into maltose, trypsin, a type of protease which breaks down proteins into peptides, chymotrypsin, also a type of protease which breaks down proteins into peptides, carboxypeptidase, which breaks down is a type of protease which breaks down peptides into its single amino acids, lipase, which is a type of lipase which breaks down lipids into fatty acids and glycerol. In the ileum, which is another name for the small intestine, maltase breaks down maltose into its single monosaccharide glucose. Sucrase breaks down sucrose into the single monosaccharides glucose and fructose. Lactase, a type of carbohydrate which breaks down lactose into its monomers glucose and galactose. And peptidase is a protease which breaks down peptides into single amino acids. just to note, once the large molecules have been hydrolyzed into single monosaccharides, amino acids, fatty acids and glycerol, they are then small enough to be absorbed from the small intestine into the blood. Just notice there's a typo there. They are then carried to different parts of the body. Then they can be used to build up again into larger molecules, which are incorporated into the body tissue as like proteins, or they can be used for processes within the body. So if it was glucose, it could be used for respiration. So using it for another purpose or building it up, using it for another purpose is called assimilation. So that's the end of the introduction to digestion.